21 types of amino acids need to be produced. In that, some of the amino acids will be coded by more than one codon. Therefore, the initiator or start codon is AUG and the stop codon is the UAA, UAG or UG. DNA very slowly it tends to mutate. Mutation, if it happens in a faster rate, then it can cause a lot of problems. So now here entire frame itself got shifted, right? So this is called as frame shift mutation. Hello everyone, a warm welcome to this session for class 12 CBSE. I am Dr. Divya, Biology faculty. So in this session of chapter 5, that is molecular basis of inheritance, we will learn about the genetic code. So what is the importance of genetic code and how mutations will occur is what we will see in this particular session. Let's learn about the salient features of genetic code. So before moving to that, I need you to remember that the genetic code always should be written in triplets. So before that, we shall recall what nucleotides are, right? So in your first PUC or in your class 11th, you would have studied about nitrogenous bases, you would have studied about nucleosides as well as nucleotides. So you know about it, right? So just brush up. So we know that in DNA, the nitrogenous bases that are present are adenine, then comes guanine, and then cytosine, thymine. Adenine, always base pairs with thymine with a double hydrogen bond. Guanine, always base pairs with cytosine with a triple hydrogen bond, or in both cases vice versa, that is thymine with adenine and cytosine and guanine. So this is the basic rule in a DNA, and these are the nitrogenous bases that are present. So remember this because we need to know about these nitrogen bases throughout. So talking about the salient features of genetic code. So the genetic code are always in triplet. So when I say triplet means tri means 3, right? So they occur in 3. So they can be in the form of AUG, UAA or CAA for example, or UAA for example, like this, right? So always they occur how? In the form of 3. So this is one genetic code, or that is one gene, this is one more gene, this is one more gene. So that is how genetic code occurs. So now you can ask me, why have I not written T there? Why we have not written T is because in the previous session, if you remember, we had learnt about transcription, right? So wherein whatever nitrogenous bases are present on the coding strand of the DNA, they get translated onto the mRNA strand, right? So therefore, mRNA means it is a RNA. So in RNA, there is no thymine. So in RNA, instead of thymine, what is there? It is uracil, right? In the case of RNA, it will be a uracil. So that is why when we write a genetic code, there is no thymine there, because DNA's function is only to transcribe the genes onto the mRNA. That way also I can put it, right? So therefore, because uracil is present in RNA and there is no thymine, it is AUG, AUA, UAA, so like that. So thymine will not be there. So that is how triplet codons are represented, and there are about, so like this, in different combinations there are totally about 61 codons that are present, or 61 triplet codons that are present, and these 61 triplet codons, they code for different amino acids. So in a body there are 20 to 21 types of amino acids like lysine, tyrosine, valine, leucine, all this you have studied in your serine first PUC as well as your 11th, right? It's the same thing. So these amino acids, from where do they come from? As such, randomly will amino acids get produced in a body? No. These amino acids are nothing but produced by the genes. So these genes, that is 61 different types of genes or triplet codons, they produce different types of amino acids, and among these there are 3 triplet codons. There are 3 triplet codons which do not code for any of the amino acids, and Therefore, they are called as the stop codon. So, amino acid chains needs to be formed because at the end of the day, our body produces proteins. So, how the proteins will get produced? So, we'll learn about that in the coming session. Okay. So, you can have a look at the next session. So, in the previous session, what we had learned that in transcription mechanism, the genes or the nitrogenous bases that are present on the DNA strand, it gets copied or transcribed onto the mRNA strand, and in prokaryotes it is a simple method of transcription, whereas in eukaryotes it is a complicated method because in eukaryotes they have both coding sequences and non-coding sequences. So, have a look at the previous session, you will understand, right? So, now they are copied onto the mRNA. Now, on the mRNA, whatever nitrogenous bases are there, they are in the form of codons, wherein 3 nitrogenous bases will form 1 genetic code. Next 3 will form 1 genetic code. That is what I have shown there, right? So, that is how they tend to form. Now, they tend to each of these genetic code, they produce one amino acid, and based on what type of amino acid. So, one protein can be made up of 20 different amino acids. So, these chains of 20 amino acids will result in one protein which will get synthesized. Say for example, our hair has keratin, our nails has keratin. So, that keratin, to form keratin is a protein. Is it amino acid? No, it's a protein, right? So, to form that one protein, chains of amino acid needs to get formed, because at the end of the day, proteins are nothing but chains of amino acids. From where will we get that amino acid? It is from these genetic codes. It is from the gene. So, that is how they produce. In that, there are 3 types of genes, or there are 3 genes. Out of the 61, 58 codons, they code for amino acids, but the remaining 3 that are there, they do not code for any amino acid. So, therefore, they are called as the stop codons. Now, they are called stop codons because amino acid synthesis can take place up till a certain codon. There needs to be stopped. Throughout the mRNA strand, can it continue coding for amino acid? No, it has to start at a particular region, like how we had learnt in transcription, initiation region or the origin of replication region, or we had learnt in replication also, right? Origin of replication and the termination region. So, here there has to be a region where it starts. T will not be there. A, U, G, C, A, C, G, U, G, A. So, maybe from here the production or the formation of the amino acids will start. So, it will go on forming amino acid up till where? Up till here. Here it will stop. Why it will stop here? Because stop codon is there. Understood? So, till there the amino acid formation or chains of amino acid will get formed. So, this is the function of the stop codon. So, there are start codons as well as stop codons here. In this, the stop codons that are there, these 3, that is UAA, UAG and UGA, they do not code for any amino acid. Remember that, it's important for MCQ. So, what are the 3? UGA, then comes UAA and UAG. So, these are the 3 which acts as the stop codon. So, moving further, some amino acids are coded by more than one codon. So, 21 types of amino acids need to be produced. In that, some of the amino acids will be coded by more than one codon. Say for example, to make you understand, say here, for example, this is a codon. It codes for phenylalanine, PHE, which is an amino acid. Now, again, there is one codon, UUC, which again codes for phenylalanine, right? Isn't 2 different codons coding for the same amino acid here, right? So, out of the 61 codons, there are few codons which code for the same amino acid. So, the codons might be different, but the amino acid produced out of it will be the same. So, next. Hence, the code is degenerate, and the codon is read in mRNA in a contiguous fashion. There are no punctuation. So, there are no punctuation. As I wrote here, have I written it as A, U, G, C, A, C like that? No, we write it at a stretch, right? So, these are the salient features of the genetic code. So, next, moving further. The code is nearly universal. So, the code is nearly universal. For example, from bacteria to human beings, UUU will code for phenylalanine only. So be it a prokaryote or a eukaryote, the genetic code which codes for phenylalanine will be UUU only. So whatever, it's just that the arrangement of the genetic codes in each organism, in each species of organism will vary. But the genetic code will code for the same amino acid in all the organisms. Say for example, UUU codes for phenylalanine in bacteria as well as humans. UUA codes for leucine in bacteria as well as humans. So like that. So valine, GUU codes for valine amino acids in all the organisms. So therefore, it is understood that whatever 61 different triplet codons are present, each of these code for a particular amino acid, or 2 or more codons can code for the same amino acid, and universally they code for the same amino acid throughout in all the organisms. So next, moving further, AUG has dual function. So AUG, it codes for methionine, the amino acid methionine, and also it acts as the initiator codon, or we can also call it as the start codon. If you remember, in the previous slide, I told you there are 2 types of codons. Out of the how many totally? 61 codons will be present. So 61 codons. In that 61 codons, 58 they code for amino acid. 3 they do not code for amino acid, and hence they are called as stop codon, because they stop the formation of amino acid at a particular codon. So we call it a stop codon. In that stop codon, what are the stop codons? UAA, then UAG and UGA. UAA, UAG and UGA. Right? So here, and out of these, so 58 code for amino acid. In that 58 triplet codons which code for amino acid, there is one codon which acts as a start codon or an initiator codon, and it codes for an amino acid. So in that 58, there is one codon which we call it as AUG. So that AUG, it codes for the amino acid, it codes for methionine, Along with that, it also acts as a start codon or a initiator codon, meaning wherever AUG is present on the mRNA strand, there the coding for the amino acid will start, and wherever either UAA or UAG or UG is present, up till there the coding or the formation of the amino acid by the triplet codon will keep on taking place, and once UAA, UAG or UGA reaches, there the coding for the amino acid will come to a halt or it will come to a stop. So therefore, the initiator or start codon is AUG and the stop codon is the UAA, UAG or UGA. So I hope you have understood about this. So this is about the genetic codes, the salient features of the genetic code. So next we'll talk about mutations and genetic codes. So we all know that the transcription process will happen wherein whatever nitrogenous bases are present on the DNA strand, they get transcribed onto the mRNA strand, and in the mRNA strand, each of these nitrogenous bases, they form triplets, that is 3 nitrogenous bases, they code for one particular amino acid. All that is we know. But sometimes there are some mutations. Mutations are nothing but alteration that can happen in the genetic sequence. So here we all have the same, say for example human beings, we all have the same arrangement of genetic sequence, and it should be like that only. Now if any mutation or alteration, maybe a extra genetic code can get added or an extra nitrogenous base can get added. Say for example, I'll just make it clear for you by writing the DNA strand. Say for example, this is the DNA strand. So I'll write a gene. This is a DNA strand. Okay. DNA A, T, G, C, A, C, G or A, G, G, T, A, C, T, for example. So for this mRNA strand, we have studied that in the previous session. For this, mRNA strand needs to get formed, right? mRNA strand. So on this mRNA strand, we'll have U. Will it have T? No, right? Because RNAs do not have thymine. Okay, so you'll have uracil, then comes adenine, then comes cytosine, guanine, uracil, cytosine, cytosine, adenine, uracil, guanine, adenine. So this is how it will form, right? So this is how it should be. Say for example, now this is the sequence. So we have UAC is one triplet codon, or it is one gene. GUC is the second gene here. CAU is the third gene here. UGA is the fourth gene here, right? So moving back, UAC. Now see here, UAC will produce what? Can you search for UAC here? Yeah, UAC. What? Tyrosine. So UAC, going back, UAC will produce what? The amino acid tyrosine. Chains of amino acids are being produced. Now GUC, let me search for GUC here. So GUC, valine. Got valine. So moving further, so GUC will produce what? Valine. So like that it keeps on, or CAU. Further, we'll go back. CAU. Histidine. And UGA is the stop. Okay. So UGA is the stop. So till here amino acid has been produced. Now these three amino acid, they can form or more chains. I'm just giving you an example. They'll produce a particular protein. Now this will produce the chains of these tyrosine, valine, histidine. They'll produce only a particular protein. So now mutation has happened. Say for example, in the DNA strand itself, mutation happened. So say for example, in here, T got added in this particular place. After G, T got added. So when T gets added here it will become adenine, right? So adenine. Now the genetic code will be UAC, AGU. Will it not become AGU? So if it becomes AGU, what will happen? Now I'll see what AGU is. So AGU, it will be serine, right? So it will become serine. So now here it will become AGU, then it will start producing serine instead of valine. Will not the protein that will be produced, will it not change? So that is called as mutation. So that is why mutation is very, very important. That is why DNA very slowly it tends to mutate. Mutation, if it happens in a faster rate, then it can cause a lot of problems. There are different types of, so you understood what mutation is right now. So therefore, mutation should not happen on the DNA. Will not mutation happen? Yes, mutation sometimes can happen, but the DNA to some extent it can repair. So that is the nature of the DNA, but sometimes if it is not able to repair, it can cause a lot of problem. So some alteration can happen in the synthesis of the amino acid, leading to different protein being synthesis. So that is about mutation. So in mutation, there are different types of mutation. So the first mutation that we'll study is about point mutation. So point mutation, as the name itself says, point. Point meaning only one nitrogenous base will get altered there. Okay. So point mutation is a change in a single base pair in the gene. Single base pair means any one, either adenine or thymine or guanine or cytosine. Any one, it will change. And that results in the change in the amino acid residue glutamate to valine. Say for example, here I told you here also, right? How got like one point mutation, one got added or one gets deleted and result in point mutation. So therefore, instead of valine, what got produced here? Serine. So therefore, the entire protein that will be produced itself will get altered. So that was what will happen. So glutamate to valine. So one of the best examples for point mutation is Mendelian principles or principles of inheritance in we had studied. So it will result in sickle cell anemia. Sickle cell anemia. We have studied that in the previous classes, that is in the previous chapter. So it results in a disease which is called as sickle cell anemia, mentioned it here. Okay, so here we will just, to make you understand exactly what point mutation is, they have given it in the form of a sentence here. So this is not how exactly the DNA on the DNA strands will not have like this. You will just have ATGC, that's it, and nothing else. But to make you understand, or if it is mRNA, we'll have A, U, G and C and nothing else. To make you understand what point mutation is, in the NCERT textbook they have given this. So see, for example, there is a sentence, Ram has red cap. So Ram has red cap. Say for example, now Ram, this one, Ram is one genetic code or one gene, has is one second gene, and red is third gene, cap is fourth gene. Say for example, it is producing protein 1, a type of protein, and I'll name it as protein A. Protein A. That is how, because Ram will produce one kind of amino acid, has will produce one, red will produce one, cap will produce one. So therefore, it will produce a particular protein. That chain of amino acid will produce a particular protein. Let's say protein A. Now here point mutation has happened. Can you see here? One B came in between S and R. In the sequence between S and R, one B is there. Now, will it not change? Ram will become one genetic code, has will become the second genetic code, BRVE will become the third genetic code, because in between in this place B came and settled there. It's point mutation, that is one of the nitrogenous base extra got added there. So, and DCA will become the fourth protein. Now, will it produce, now didn't the chain of amino acid vary? Now, will it produce protein A itself? No, it will produce some other protein. So, that is called as point mutation. Moving further, we'll talk about frame shift mutation. Frame shift mutation means here, if two or more nitrogenous bases get added, what will happen? So, here again, they have given Ram has red cap. So, Ram is the first genetic code, then comes the second, this is the third, and this is the fourth. So, as I told you, it will produce a protein. We named it as A, right? So, protein A it will produce. Now, here, before red, RED, B and I, what got added there? The sequence B and I got added. Now, Ram will become 1. This will become the second genetic code. BIR will become the third. This will become the fourth, and this so on. So now here frame, entire frame itself got shifted, right? So this is called as frame shift mutation. Here insertion or deletion of one or two base pairs will happen. So there one base pair insertion will happen, which will result in point mutation. Here either insertion or deletion of one or two base pairs, it changed the entire reading of this frame, right? So Ram has Bir EDC AP, it became, didn't it entirely change the reading there? So that is what we call it as frame shift mutation. And here again, say for example, here 3 sequences got added, B, I and gene. Ram 1, this is second, now this is third, this is fourth, fifth. Ram has big red cap, didn't it just shift, right? See here, actual sequence in the third genetic code, it was supposed to be RED. But what happened here? It became BIG, right? Because extra nitrogenous base pairs are getting added there. So that is why. Now see here, if deletion happens, what will happen? We will see. So here deletion, Ram is there, has is there. Then EDC, what they did, they deleted R. So if R is deleted, what will happen? Or R is deleted. R got deleted, what will happen? So if R got deleted again, Ram will become the first genetic code, then this will become the second, this will become the third, and this will become the fourth. So like that. Again here also, say for example, again here Ram first, second, then here E also got deleted. Now R is also deleted, E is also deleted, right? So R also deleted, that's why I put a cross mark here. E is also deleted. Now it will become Ram has DCAP. Now here D, red, complete red is only deleted. They've deleted R, E and D. It got deleted. Ram has cap. Only that is remaining. So entire frame got shifted. So the mutations are the genetic code or the sequences are getting shifted. The genes are getting shifted. So that is why we call it as a frame shift mutation. So these are the two different types of mutation in the genetic code. So this was about the session wherein I hope you found it quite interesting, because we learnt about genetic code, how the sequences will be there, and in that 3 nitrogenous bases will form the genetic code, which is the triplet, and how their mutation, either insertion or deletion, what will happen if it gets inserted or deleted, or what happens at point mutation and frame shift mutation. So this was about this session. So in the coming session, we'll learn about the mechanism of transcription. So we had learned replication in the previous session of prior to this. In the coming session, we will learn about transcription, that is tRNA. So what is in transcription? tRNA, or the transverse RNA, plays a very important role, and it is called as the adapter molecule. Why is it called the adapter molecule? What is its function? We'll look into that, and we'll learn about the steps involved in the process of translation. So see you in the next coming session with these two topics. Thank you.